Hello, everyone, and welcome to the Anti-Doping Podcast, brought to you by the Partnership for Clean Competition. I'm your host, Dr. Marie McNeely, and today we are joined by our guest, Liesl Janssens. So, Liesl, welcome to the show today.
Thank you. It's been a while, and I'm very happy to be back.
We're delighted to have you with us today, listeners. Liesl is a PhD student at the Laboratory of Toxicology at Ghent University. And if you haven't listened to our interview in episode 59, you should definitely check it out. And today, Liesl is going to give us an update on her project on hypoxia-inducible factor, or HIF, stabilizers. And she's also going to discuss characterization and detection of cannabinoids, opioids, and psychedelics using activity-based assays. So, Liesl, we are looking forward to talking about all of these things. How are you doing today?
I am very good. It's a Friday. Looking forward to the weekend and first this nice interview with you. So looking forward.
Oh, well, we are excited to learn more and to get an update on all the amazing things you've been working on over the years. So for listeners who might not have met you yet, haven't listened to episode 59, can you start by telling us a little bit more about yourself and your background?
Well, I studied pharmaceutical sciences, first bachelor at Ghent University in Belgium. Then I continued with a Master of Science in Drug Development at the same university and faculty. And in those studies, I've really created a profound interest in cell biology, biotechnology, and toxicology in particular. So after having contact with my professor of toxicology at that point, Christoph Stove, I was invited after my Erasmus program to discuss with him a possibility of doing a PhD. And after finding a project that is based on all those topics that I like so much, here I am right now doing a PhD on all of that.
Well, I think it is a really exciting project that you've identified with Christoph and that you've been making amazing progress on. But I guess what made this particular project on HIF stabilizers interesting to you? Why did you choose this particular item for your PhD?
I would say definitely the link between real life issues and cell biology. As cell biology, the knowledge of that and the techniques in that, like biotechnology and stuff, I really love working with that and love learning about that, but I wanted to do something application-minded with it. I never wanted to do very fundamental research, gain fundamental insights on signaling pathways, for example. I really liked my research to be very comprehensible to the bigger public. So the fact that I can join fundamental interest in cell biology and use it as an application tool to address real-life issues, that was something that interested me a lot. So therefore, I started a PhD on it.
Absolutely. And I think that's one of the really exciting things about anti-doping research in general is a lot of times you can see that research that you're doing in the lab having an impact in the real world on a relatively short time frame, which I think is absolutely amazing.
I completely agree. That was also kind of my end goal starting 4 years ago, that I wanted to deliver something that might be applied somewhere in the near future.
Absolutely. And to give our listeners a little bit of an overview about HIF stabilizers, why is it important to investigate these HIF stabilizers within the context of anti-doping?
I guess HIF stabilizers are a very smart, so to say, way of circumventing the use of EPO injections. They increase your own body's production of endogenous EPO instead of injecting recombinant EPO. And that endogenous EPO is not distinguishable from any other EPO in your body, obviously. The fact that also a HIF stabilizer can just be taken orally, it's like swallowing a pill, makes it very easy for athletes, I think, to take that step. So definitely in the context of clean sport, we should look for them because they increase oxygen transport. And of course, increasing oxygen transport increases endurance and increases performance in the end. Maybe that's also relevant for listeners that it's kind of a chemical alternative for high altitude training. Because what happens at high altitude training, you train with less amounts of oxygen present in the air. And what that does to your body is literally also stabilizing HIF. So HIF stabilizers are kind of the chemical alternative for that. So definitely, if we want to protect clean sport, we have to look for them. But I think also from a perspective of ensuring the athlete's health, it is very important to take them in mind. The HIF pathway itself is associated with cancer. I have no knowledge on real cancer-related side effects that have been seen in clinical studies of HIF stabilizers yet, but I do know that quite the opposite of HIF stabilizers, which are then HIF inhibitors, are actually used as cancer therapeutics. Because high levels of HIF, which is of course also obtained or attained when stabilizing HIF, high levels of HIF in cancers are associated with a very bad prognosis. So in theory, HIF activation effects can worsen cancer through, for example, metastasis, metabolic programming, immune evasion. As I said before, there is no real knowledge yet if that is happening with intake of HIF stabilizers, but it's definitely a concern that I would upheld for myself if I would dare to take those drugs.
Absolutely. And I think maintaining the health of the athlete is a huge part of this anti-doping movement. You don't just want to ban all these substances for the sake of sport. It's also for the sake of protecting athletes, which I think is critically important. And I know when we last spoke, after more than a year of hard work, Liesl, you had developed this process for activity-based bioassays to detect HIF stabilizers. So can you describe how this assay works in general?
Let me then maybe start with activity-based aspect of it, because we measure activity, which is quite different from normal techniques that are used in anti-doping control, such as mass spec. Because with mass spec, you look at typical or specific chemical structures, so specific chemical molecules. But we are not looking for that. We are looking at the effect of those molecules. So in fact, anything that has that effect will be detected by the assay. And anything that has that effect would then also have to mean that something is wrong. A HIF stabilizer, for example, they stabilize HIF. So that's an effect of that is that more HIF, in particular the HIF alpha or HIF A part, will be present. But to have a performance enhancing effect, HIF must act as a functional transcription factor, because the acronym stands for Hypoxia-inducible factor, which is a transcription factor. It makes sure that genes are expressed, for example, the gene of EPO, which then has the performance enhancing effect through more blood cells that are running through your blood flow. So to act as a functional transcription factor, the A part that is being stabilized needs to interact with the B part. And that interaction is what we measure. That is the effect or the activity that we use as a detection mechanism. So an increase in interaction with the B part would mean that something wrong is going on there in a sample or in a powder, doesn't matter really. Anything that is unnatural, because normally if no HIF stabilizer is present on what you apply on the assay, then there would not be that much of an interaction with the B part. And how do we measure that interaction? That is based on coupling two subunits of a molecule that can emit light, but that molecule can only emit light if those subunits are brought together. And that is being done by the interaction of part A and B of HIF. So light is emitted if something wrong is going on in a sample or a powder, for example.
That makes sense. And I appreciate you kind of breaking it down, because I know there's complex science behind it. You presented it well in a way that's digestible here. And we mentioned that you've been working on this project for a number of years. So can you give us an update on some of your progress, or maybe the milestones that you've reached on this project over the past 2 years?
So the assay itself relies on the expression of these coupled proteins of that molecule that emit light and the HIF parts A and B. So previously, that DNA that we made for that, we had to introduce it into cells so that cells would express it, and then we could use those cells as a tool for the detection. But we always had to do that every single time for the experiment, because we used transient transfection. So for every experiment, we needed to introduce that DNA into cells. And doing that over and over again brings a lot of variability, which is quite logical because you can't control all of the factors every time. And to address that problem, or that high variability that is associated with transient transfection, we made a stable system out of it. We infected the cells with a virus, and that virus brought that exact DNA that we wanted into the cells, into the genome of it, so that now the cells, and they are constantly expressing what I want them to express. And that inherently brings that the assay is now way less variable and also more sensitive, because it's a more constant amount of proteins that is in there. What another update that I can give you is that I have been continuing of running HIF stabilizers on the assay, and there has not been one HIF stabilizer that did not give a signal in the assay. So that is quite good from a future-proof aspect, because I can assume now that every compound that will stabilize HIF will be detectable depending on the concentration that is. But anyway, it's very future-proof. And I think then the most important, definitely from an anti-doping perspective, is that we are now running samples on the assay. We have proof of concept that we can distinguish a sample with HIF stabilizer in there from a sample without HIF stabilizer. But now the goal is to find the optimal sample preparation, and that's what we are currently working on. We want to prepare the samples as well as possible so that we can gain the sensitivity that we want in the long run.
So Liesl, in terms of sensitivity, do you have an idea of how much HIF stabilizers must be in the sampler, must have been taken to be detected? Is it one pill, or does an athlete need to have been taking them for a long time?
Well, we are currently mostly looking at sensitivity in terms of concentrations that could be present in the urine of athletes. So not really a dose or a period of time, because this way, looking at concentrations, we can determine, or we want to determine, the lowest concentration that we would be able to detect in urine samples. But maybe important to note is that we are not working on athlete samples yet, as we first want to establish a proof of concept and get an idea of the potential performance of the bioassay, definitely before applying those precious authentic samples on there. So in very practical terms, that means that I'm kind of working on my own urine, so to speak. And that might sound funny, but that is how research goes. Mm-hmm. You optimize your system based on easily available and less precious samples. So, for example, my own. Also important to note is that I am not taking HIF stabilizers myself, obviously. I'm merely using my own urine and I spike it externally with HIF stabilizers in a specific concentration so that I can see what that hypothetical concentration in a urine sample would give as a result in our assay. And that way I can see how low these concentrations can go to still be able to detect them. And I think at this point we are at 25 nanograms per milliliter, something like that. So we want to go lower still, but that is where we are at right now. Therefore, it is very hard for me to say which dose or time period that would correspond to those concentrations, as I'm artificially adding the doping to my urine in a tube, actually. So instead of taking those drugs myself and peeing them out, as would be the realistic scenario for cheating athletes and samples that are sent to the doping lab. So amount of pills is kind of hard to get.
Absolutely, Liesl. I think that makes sense. It sort of depends on what the HIF stabilizer is, what the metabolism looks like, and what the form that the drug would be administered in, whether it's, a capsule and what the dosage of those would be, what you're able to detect. Now, do you have a sense of the timeline for any known HIF stabilizers in terms of how long it actually takes for the body to process them and for them to be detectable in urine?
I think the best way for me to answer that question would be to refer to the publication of Liesl et al. They really investigated a real positive case for the misuse of voxelotor. In that case, the athlete had taken 100 milligrams of voxelotor every 2 days for 19 consecutive days. And this was in between two samplings for anti-doping control. 19 days is quite a long period, I would say, but I can imagine that would be necessary to really get the whole process running of your body producing really significantly higher amounts of EPO so that really a noticeable effect on the red blood cell count and on the performance can be attained. But after this period of 19 days, that athlete was misusing the HIF stabilizer voxelotor. He tested positive in his urine, so the day after, with a concentration of 18 micrograms per milliliter. And this is a concentration that would also very easily be picked up by the bioassay. To give a sense of how long it can be detected, in that particular case, 20 days later, the athlete still tested positive in urine. But that was at a very low concentration, important information that is. So that is definitely thanks to the highly sensitive instruments that anti-doping control is working on.
Definitely. Well, I think that's reassuring that there is a reasonable detection window that you might be able to identify if someone is taking these kinds of compounds. Do you know yet if this method also works to detect some of these metabolites of HIF stabilizers that it might be broken down into in urine?
That is a very good question, and actually a very fitting one, because I'm currently looking for funds to try and study that. It is a very interesting question because if a metabolite is still active, which our assay would be able to show, then that would benefit the sensitivity. And we are currently seeking to custom synthesize reference materials of these HIF stabilizer metabolites so that we can really profile them and how active they are also in comparison to the parent or the HIF stabilizer itself. And if metabolites are active, like I said, this could benefit the sensitivity. The assay detects the combined effect. So everything that has an activity will stabilize the HIF. And I'm guessing those activities will be additive as well. And that's something that I also want to investigate. But for example, to give a link already with what we want to talk about later, the cannabinoids, there the metabolites are most cases still very active. For example, in a study that we really run or screened patient samples, metabolites only that had a concentration of 0.57 nanograms per milliliter, which is not much, was still giving a positive signal. So depending on how active metabolites of HIF are, that can be a very interesting thing to study. So it's definitely on the list for future steps.
Oh, fantastic. And Liesl, you've talked about this activity-based assay, and I think it sounds amazing. What remaining steps need to happen before this assay can actually start being used in routine doping control testing?
In my opinion, it can already be used, but this would be in conjunction with the ABP. Because, for example, referring again to that case of Liesl et al., there, right after the day after the last administration, they detected a concentration of 18 micrograms per milliliter. That is a concentration that is quite high, I must add, but that is something we would also easily, very easily be able to detect. And in that case, the ABP showed in one parameter a suspicious change. The ABPS was reaching the predicted limits, but all the other parameters were not. Still, there was nothing to be flagged because it was still in the limits of the intervariability. But in that case, such a suspicious sample would be able to be run on the bioassay. And by that, you would already be able to show that something HIF stabilizer related is in there. So the bioassay at this point can already give a definite answer if high enough concentrations are present, if the suspicious ABP, for example, would be HIF stabilizer related or not. In terms of screening, there we are not there yet. Therefore, I'm still looking for the optimal sample preparation and maybe also some other things to try and increase sensitivity even more, or maybe look for confirmed specificity of signal. But anyway, at this point, we mostly have to do something about the matrix effect because different urines give at this point different signals in the assay. And that's, of course, very difficult for a screening assay as you don't want to have too many false positives or false negatives. So screening-wise, there's some optimization to be done. Confirming-wise, or getting a definite answer if something is HIF-related or not, I think that is in the very near future. I hope so, at least.
Oh, wonderful. And in terms of the setup, you mentioned that you're using cell culture, some of these more biological-based setups within the labs. Do you think WADA-accredited labs would be able to adopt this easily, or are there some sort of overhaul of lab infrastructure and equipment that would be needed?
That is a very fair question. Working with cell cultures means indeed that you need some specific infrastructure, such as, for example, sterile cabinets, but also sterile materials and an incubator for the cells to grow in, for example. So to implement this in a routine doping lab, you would indeed need to buy some new equipment, and also the staff should be trained for working with cell cultures, which mainly entails sterile handlings. But in my opinion, this would not mean a real burden to adopting this. The things you need are not taking that much space, and the training would also not have to take that much time, as people tend to usually get a hang of it very fast. But I have to admit that I kind of take it for granted because I have been doing this my entire PhD and it just feels so accustomed. But generally, I would see this type of analysis also more from a centralized or collaborative point of view. For example, here in Belgium, we are located almost next to the doping lab of Professor Vanino. So if he would have a case like that with a suspicious ABP result that he cannot explain, for example, we would be able to run that sample or those samples that are around that sampling period on the assay and confirm or rule out that it is HIF stabilizer related or not. And we could also do that for other labs in the future if needed. In any regards, by now, I think the real strength of the assay would not lie in implementing it as a routine assay, but by being so future-proof. So keeping guard of what is circling around amongst cheating athletes and to be ready to detect any new HIF stabilizing compound, since it is such an evolving field. So at this point, implementing it in a centralized way would be the way to go anyway, I think.
That makes sense. And I think it really highlights the value of these collaborations between academic institutions and WADA accredited laboratories, where you could potentially work together so that you don't have to do a complete overhaul of the infrastructure of the WADA accredited labs.
For me, it's also nice to be working on both sides. I am working in an academic environment, but also get to see what is happening in a routine lab. So it's also, from my perspective, it's very nice to see and very interesting collaboration.
Fantastic. And I know that since our previous interview, you've actually won the Young Scientist Award for Best Oral Presentation at the recent 2022's virtual meeting of the International Association of Forensic Toxicologists for your presentation on this PCC-funded work on HIF stabilizers. So first of all, Liesl, congratulations.
Thanks.
And I'm curious, you've seen this project develop and you've had a lot of success with it. So what has it been like for you personally to see the success of this work, to see all these milestones that you're hitting and to be recognized for your excellent work?
I would say nothing less than an absolute honor. It's nice to see that my enthusiasm about the project is shared by other researchers, as it was apparently contagious during that meeting as I won the award. But it means also a lot to me that it was at the Conference of Forensic Toxicologists as well. I also work on many toxicology-related topics, which is where my main interest for attending that conference came from. And anti-doping research definitely also has a place in that field, but I would say that it's more of an outsider in that regard. For example, in comparison to real hardcore drugs like opioids or cannabinoids. So I must say that I was surprised receiving the award with my presentation on HIF stabilizers, but incredibly happy even more so, because as you said, reaching milestones, we are not quite there yet. For example, as referring to your previous question, I know that it will not be implemented in a doping control lab anytime soon, or that it's very hard to get all that infrastructure there as well. But little steps are also something to be happy about, and I am most happy with the steps I'm taking.
Absolutely. And I know, as we've alluded to, you're also applying this approach of activity-based bioassays to other types of compounds, not just these HIF stabilizers. You mentioned cannabinoids, opioids, psychedelics. So, Liesl, can you discuss what makes a compound a good candidate for this activity-based assay approach? And are there other types of compounds that maybe aren't as good of a fit to be using it?
Well, let me first give a shout out to my colleagues as well, because I'm not working on all of those compounds. I think I would not have the time to do that. I'm working mostly on the cannabinoids, and I have some very good colleagues who are working on opioids and psychedelics. Let me then come back to the fact of activity-based detection, because this is really an untargeted approach. And that's also where the good fit lies in. We are not relying with this activity-based detection on chemical structure. So you measure everything that has an activity or has the activity that you're actually measuring. So a good candidate for such a method of detection would always be a class of compounds, because you're able to detect many at once. Activity also must be easily measurable, because not all signaling pathways or not all interactions are as easy to really monitor. So that's also something that every researcher who is writing a project or a grant proposal will have to take that in mind. But when I tell about a class of compounds, that means that there are many compounds in that class. But the benefit of activity-based detection is that there also can be a high variety of chemical structures in that, possibly also unknown ones, emerging ones. As, for example, for the HIF stabilizers, this is a field that is very interesting from a therapeutic point of view. So I'm expecting way more HIF stabilizers to be studied in the next few years. So way more HIF stabilizers are coming on the list that we will need to be able to check for. And that's why HIF stabilizers in the doping field is a very interesting class to look at activity-based, because you really, with activity-based detection, you generate a future-proof method, and you can act proactive to include already compounds that maybe not even exist yet, because if those compounds would have that effect, they would be detected by that bioassay. What would not be a good fit is, for example, one particular chemical structure and no prospects of new ones in the pipeline or new ones being investigated anytime soon. Then I would not make activity-based bioassay for that. Then I would go for mass spec, which has excellent sensitivity, excellent performance. So there is no benefit in then working on an activity-based approach. What can be a problem in activity-based approaches, and that's also where it is related to classes of compounds that might not be a good fit, is that there can also be endogenous compounds that have effects on your assay. And if you're applying biological matrices like urine or blood on your assay, there might be compounds in there that also activate it, and that complicates the interpretation of the assay. In terms of doping, for example, that would be androgens. It would actually be very good if we have an activity-based assay, which already exists, gene reporter assays, but they're not really applied in routine doping control. But it would be very good in that class, because we are in doping currently struggling with the abuse of SARMs. And those SARMs are also a class of compounds with a high variety of structures. So that would actually be an ideal fit for an activity-based assay. But the problem there is that you also have testosterone, which is excreted in both men and women. And that testosterone would also have an activity in those androgen activity-based assays. So in that regard, the class of SARMs would be a good fit based on chemical structures, based on variety, based on being able to detect the activity. But you would have to come up with dedicated sample preparation. You would have to adjust your system to ensure specificity, because those endogenous compounds, well, they do play a big role in the interpretation of that.
Absolutely. And I like that you're thinking about this broadly. You don't have tunnel vision where you're focusing so much just on these HIF stabilizers. You're thinking about all of these different applications. And you mentioned that a lot of your research now is focusing on these cannabinoids. And as our listeners may know, all natural and synthetic cannabinoids are actually banned by WADA in competition. And last year, Liesl, you published a paper examining the performance of an activity-based method for detecting new synthetic cannabinoid receptor agonists, or SCRAs, things that don't even exist yet. So can you start by describing what these synthetic cannabinoid receptor agonists are and why this untargeted approach is particularly useful for detecting this class of compounds?
Synthetic cannabinoid receptor agonists, or in short, SCRAs, as I call them, are a class of designer drugs. They are intended to mimic the effects of cannabis, but in fact, they surpass the effects of THC in cannabis enormously. And it is a typical class to detect activity-based because there are so many chemical structures in that class. I think there are over 200 designer drugs of cannabis already identified since 2008 only in Europe.
Oh, wow.
That's why this is a very good fit for an activity-based assay. Straws in general are, as I said before, surpass the effects of the original cannabis, which means that way lower doses are required to have already way stronger effects. But that also means that it's more dangerous, way more dangerous, and the risk of intoxications is very high. The synthetic variants of this cannabis were originally intended and investigated as therapeutics to treat pain and nausea, stimulate appetite, and enhance mood. But by now, they are very well or very frequently and extensively synthesized chemically in clandestine laboratories for the illicit recreational drug markets. And as I mentioned before, there is a large structural variety in this class because these clandestine labs are constantly altering the chemical structures of existing compounds to circumvent legislation. And that led to a real cat and mouse game for the past decades between updating legislation, updating mass spec methods, and the coinciding continuous emergence of novel analogs. Therefore, a perfect candidate for activity-based detection because we want to be able to detect all of them, the ones we know about and the ones that are still coming out.
Absolutely. And I know you actually compared your activity-based approach to the sort of traditional gold standard of liquid chromatography coupled to high-resolution mass spectrometry for detecting these straws. So, Liesl, what were your findings in terms of how your activity-based approach performed to the gold standard?
It was very well, actually, closely matched the gold standard. But I have to say that this is really class-based and activity-based, bioassay-based. This is not to be directly extrapolated to other activity-based assays, of course. But in terms of the cannabinoids, we reached a sensitivity of 94.6% and a specificity of 98.5%. Which is very good. Mm-hmm. The only samples I think that we missed were samples where only metabolites were in there and they were at very low concentrations and they were just the metabolites that are less active or where the difference between activity parents and metabolite was very big. The only sample that we missed were quite logical that we would miss them. Let's say it like that.
Absolutely. And I think that's really promising in terms of the capability of these methods for detection. And I think that is amazing, like I said, to be comparing to this traditional gold standard and to see such high performance. Were you surprised?
Well, there was a study before as well from my colleague Liesl Janssens, and there it was already very good. But we actually lowered the sample volume because the less sample we have to use for a screening assay, the better, of course. So we lowered it, I think, by half, and we still had these results. So yes, I was surprised, but very happy nevertheless.
Oh, definitely. And you also developed methods using actually machine learning to facilitate screening and interpretation of this output, because I know the output can be sort of a big mess of data that you have to wade through to understand what these results mean. So can you talk a little bit more, Liesl, about how this works?
Well, before applying machine learning, as you said, you have a big bunch of data and you have to look at it. And how it works for activity-based detection is that you compare the activity profile. So basically, we measure a kinetic profile for 2 hours. And in that hour, activation of the receptor or stabilization of HIF is happening. So you compare those receptor activation profiles with receptor activation profiles of the blanks, where not much receptor is being activated. But up till now, this always has been done visually. So you really look at both profiles, you compare them, and based on that, you make a decision, this is positive and this is not. But as you can imagine, this is a very subjective process. So first, we tried to objectify it and standardize it a bit, like how should we interpret such a profile? And so we made a decision tree for that, which is in that same publication. And that worked better. The consensus between two individuals looking at such profiles is better. So that's definitely already an improvement. But still, you have to look at curves. And even the slightest difference can mean that there is a metabolites at low concentrations in there, for example, which was the case in some samples. So therefore, we really thought objectifying and standardizing even more so that the human component is extracted from it would be a benefit. Also from if you want to transfer that method to another lab, for example, it is easier if you can just transfer the machine learning method as well so that no people have to be trained to look at those data. And therefore, we built a computative model with another department of Ghent University. And that model then was trained based on our input. So we told that model, this is how a positive sample in our opinion looks like. This is how a negative sample looks like. And then the model trained itself based on that input. So based on a supervised learning approach for the listeners who are into machine learning. And eventually that model predicts a score. And that score would say, how likely is it that this sample would be positive or negative? And then based on how sensitive or how specific you want your assay to be, you can set your threshold yourself. And that determines how many false positives and how false negatives you will let through. And then based on that threshold, the computer just gives you a yes or no answer. It says, yes, this is positive, or no, this is negative. That means that applying such a machine learning model is a huge reduction in workload. I think I spent hours, and my professor as well, we spent hours on looking at the data and then having a discussion how we should interpret that. So now it's not so many hours of data analysis anymore. You just put the raw data in there, the profiles, the kinetic profiles, that is, and then the output is yes or no. So I think that if we would be able to implement that on a regular basis, that would be great in terms of hours spent on data analysis.
Absolutely. I think this helps make, as you alluded to, the interpretation process so much faster and also so much more consistent. Like you said, different raters might be thinking about these samples differently and not necessarily applying the tree the same way when they're making a decision. So this is a great way to just make that more consistent across labs and across individuals. And I know the PCC, Liesl, is so excited to be able to support some of the work that you're doing, but how have your interactions with the PCC maybe impacted or enhanced your research?
As I mentioned in the beginning of this interview, I have a real interest in cell biology, and the activity-based detection line that Professor Stover was running in his lab really intrigued me. And he and Professor Van Eno from the DOCO lab, they had a discussion about the HIF project, and that HIF project was made possible through funding of the PCC. So I think it literally has an impact on me doing this PhD right now. But I must say that I also have felt a lot of support of the PCC. Acknowledgement of my tea afterward, for example. I think they spread it on Twitter even. I think they made a post out of it. Also spreading the research. If my first publication came out in analytical chemistry, I think they also spread it on Twitter, which is highly appreciated. And also through communication, I can feel a lot of support. And it's nice to know that an organization that is funding doping research believes in this. That means a lot on my own motivation as well.
Absolutely. And I know your career is progressing. And when we last spoke, you were in your second year of your PhD program. Now you are in your fourth year. So, Liesl, have you started thinking yet about the next stage of your career and what you want to do after you finish your PhD?
I have, but it's a work in progress, I must say. I'm really passionate about research on using cell biology to our advantage, being as means for an activity-based bioassay for detecting drugs or doping, as I have studied during my PhD or as I am studying right now. But it could also be as a diagnostic tool or maybe even cell therapy. Anyway, it still intrigues me very much. And in this regard, I am slowly starting to dream about doing a postdoc to further exploit the possibilities that cell biology and biotechnology have to offer us. And I also dream of continuing that career abroad, as internationality has always fascinated me ever since I went on Erasmus to write my master thesis there. And I am on the lookout for interesting possibilities in this context. But as I said, it's a work in progress because I currently don't have that much time to actively look for it, as I still have a PhD to finish and I'm not quite done yet. I still have many things I want to try out before ending this. So I have thought about it. I am thinking about it, but it's not done thinking yet.
Absolutely. I think there are a lot of exciting possibilities and wonderful directions that you could go. And I know anti-doping science in general, it's a very international community, in science kind of more generally as well. So we hope that you can eventually fulfill that dream of training abroad. And we've talked about a lot of different things. We've gone into some of the details of your research, but I think there's so much more for listeners to learn. So if they do want to learn more about you and your work, Liesl, what is the best way for them to do so?
I would obviously guide them to my publications, which is easily to be found on PubMed or Google Scholar or website Web of Science. I also have a Twitter account, and usually when something new is being published, I will post it there. So people can definitely follow me there, and hopefully they are also interested in the work. And that will be the best way to get to know about my work.
Wonderful. Well, listeners, definitely get connected with Liesl on Twitter. Check out some of her publications. And as you may know, listeners, Liesl was one of the winners of the PCC's virtual poster session in 2021. So you can also find a video of her presentation as well as videos from other presenters on the PCC's YouTube channel. And Liesl, it's been such a pleasure to have you with us today. Thank you so much for joining us again.
Thank you for having me. It was 100% my pleasure.
Well, we have enjoyed learning from you today. Listeners, it's been great to have you here with us as well. And if you haven't already, it would mean a lot to everyone at the PCC if you could rate our podcast and leave a review on your favorite podcast platform. And listeners, please note that the Anti-Doping Podcast features a wide variety of clean sport experts, and episodes may include statements and or viewpoints that do not necessarily reflect the views or values of the PCC. Thank you, and we look forward to seeing you on the next episode of the Anti-Doping Podcast.